secretions. Introduction. Digestion and absorption of food requires GI secretions with enzymes and GI motility, which is regulated by enteric nervous system and GI hormones. Layers of GI tract. You have your mucosa innermost, then muscularis mucosa, outer surface area, no role in motility. Then submucosa, which has submucosal plexus, regulates GI secretion, also known as Meissner's plexus. Then you have mandibal plexus, known as Orbeck's plexus. Then GI motility, you have before submucosa, you have muscularis propria. Number two layer. Sorry, after submucosa, you have muscularis propria, longitudinal muscle, and circular muscle, and the outermost is serosa. Daily water turnover in GIT. Total water in GIT is almost 1 liter. So total water in GIT 9,000 ml per day. The absorption 8,800 ml per day. Excretion in stool 200 ml. Ingested fluids 2,000 ml per day. GI secretion saliva 1,500. Stomach 2,500. Exocrine pancreas 1,500. Bile 500. Intestine 1,000 ml. Jejunum 5.5 liter, helium 2 liter, colon 1.3 liter. Saliva, digestive and protective functions. Properties of saliva pH 7 hypotonic to plasma. Three major salivary glands, parotid duct and parotid gland, accessory parotid gland. Parotid gland is the largest. This is the polymandible. This is submandibular gland. This is sublingual glands. An opening of submandibular glands, Wharton ducts. Few minor salivary glands, Weber's gland in tonsils, Weber. Von Ebner's gland in base of tongue. Functions of saliva. Function of enzyme involved in digestion. Types is by salivary amylase and chylin. Cat's bilingual lipase by von Ebner's gland. And protein enzymes. Lubrication to prevent injury to buccal mucosa by dry foods. Protection, immunity, comfort through secretory immunoglobulin A, lactoferrin, lysozymes, which are antibacterial in nature. Saliva. Functional unit of salivary gland is salivary acinus and duct acinar cells. Primary secretion similar to plasma. Duct cells. Reabsorption of sodium and chloride, secretion of potassium and bicarb, secondary secretion. During high flow rates, decreased time of resorption, secretion has increased sodium and chloride. Regulation of secretion. Salivation is an unconditioned reflex. Regulation by autonomic nervous system is sympathetic system and parasympathetic system. Sympathetic system is increase on secretion of thick saliva. Parasympathetic profound increase in secretion of thin watery saliva. Bradykinin, vasodilation, increased blood flow to salivary glands, increased secretion, physiological factors that affect saliva secretion, increase in sight, smell, and thought of food, decrease in sleep, fear, and fatigue. Innervation of salivary gland. parotid gland, otic ganglion, coming from cranial nerve 9, sublingual gland, coming from submandibular ganglion, cranial nerve 7. Increased chances of injury to cranial nerve 7 during parotid surgery. Abnormalities of salivary secretions. Saliva. Absent secretion is called a diarrhealism. Decreased secretion, xerostomia, dry mouth, commonly seen in mumps and Sjögren's syndrome. Diarrhealism is also seen in mumps and Sjögren's syndrome. Associated abnormalities, injury to facial nerve during parotid surgery, during regeneration. Innervates sweat glands, increase salivation, increase swelling. Innervates lacrimal gland, increase salivation, increase lacrimation. Crocodile tears, gustatory lacrimation. Hmm. That is injury to facial nerve during parotid surgery. Gastric secretions. Gastric secretions, volume 2.5 liter per day, pH 1.5 to 3, most acidic secretion. Gastric glands, flask-shaped gastric glands. This is the mucosal surface. Surface cells produces bicarb-rich mucus for acid neutralization. Trifol peptides confers immunity. Neck cells, stem cells for gastric glands. Parietal cells produces HCl acid. Intrinsic factor of Castle for B12 absorption. Enterochromaffin-like cells reduces histamine, increased acid secretion. Enterochromaffin-like cells produces histamine. Chief cells produce pepsinogen, and hence enterogastric cells, G cell, produces gastrin, increase acid secretion. Note, in pernicious anemia, autoantibodies to parietal cells, destruction of parietal cells, atrophic gastric mucosa, ratio hydra, increased acid secretion, anemia, decreased B12 absorption. Acid production in parietal cells. You have your A is proton, potassium, sorry, hydrogen ion, potassium, ATPase, proton pump. B is your chloride bicarb exchanger. C is chloride channel. And Ca is carbonic anhydrase. Postprandial alkaline tide, increased HCl production after meal, increased bicarb to blood. Postprandial alkaline tide. Acetylcholine vagal synapses, coffee, alcohol, calcium ingestion. Histamine acts on H2 receptors, gastrin on cholecystokinin B receptors, acetylcholine on M3 receptors, factors that decrease excess acid, negative feedback, somatostatin universal inhibitor, prostaglandin, cholecystokinin, secretin, calcitonin. Functions of gastric acid. Functions: digestion of carbohydrates, no enzymes. Fats, gastric lipase, proteins, pepsinogen activated by HCl, pepsin, and renin is found in cows. Protection, increased acidity, microorganisms killed. Phases of gastric acid secretion. Percentage of gastric acid secretion. Cephalic phase, gastric phase, intestinal phase. Cephalic phase, in absence of food, 20-30%, initiated by sight, smell, or thought of food. Gastric phase, 60-70%, when food enters stomach, enteral G cells release gastrin, release acid secretion. Intestinal phase, due to products of protein digestion. Intestinal G cells release gastrin, increase acid production. In gastric phase, gastrin coming from enteral G phase, and in intestinal phase, gastrin coming from intestinal G cells. Exocrine pancreatic secretion. Head of pancreas, common bile duct, pancreatic duct, tail of pancreas, lobules. SNR cells secrete digestive system. Pancreatic duct cells secretes hormones. Pancreatic duct exocrine cells secretes pancreatic store nutrients. 1500 ml. pH is 8.3. Bicarb is more. Function: digestion of carbohydrates, pancreatic amylase. Fats, pancreatic lipase assisted by colipase. Proteins, zymogen, inactive enzymes, trypsinogen, chymotrypsinogen, procarboxypeptidase. Activation of zymogens in small intestine. Trypsin for protein digestion activates or stimulates chymotrypsinogen and procarboxypeptidase to become chymotrypsin and carboxypeptidase. Trypsin itself is coming from trypsinogen, which is activated by enterokinase. Note: inflammatory process, premature activation of zymogens, acute pancreatitis, autodigestion, regulation, hormone regulation, cholecystokinin, pancreozymin, constriction of gallbladder, increased pancreatic enzymes in secretion. Secretin, nature's antacid is secretin. Nature's antacid is secretin released from S cell in intestine on stimulation by acid chyme. Increased bicarb secretion into pancreatic juice, alkalinity of pancreatic secretion for acid neutralization. Note: secretin, first hormone to be discovered, decreased enzyme concentration. Mechanism of bicarb secretion. Pancreatic cell. A is chloride bicarb exchanger. Chloride bicarb exchanger situated at label A. CFTR is cystic fibrosis transmembrane conduction regulator. Chloride channel opening. Mutation: cystic fibrosis. Cystic fibrosis transmembrane conductance regulator. Coming to biliary secretions, liver synthesis of bile 97% is water. Gallbladder storage and concentration of bile 287% water. Bile is produced by the liver and stored in gallbladder, liver. Functions of bile, digestion and absorption of lipids, excretion of bile pigments and cholesterol. Constituents of bile, primary bile acids in liver, folic acid, chenodeoxycholic acid, intestinal flora, secondary bile acids in intestine, deoxycholic acid, lipocholic acid, conjugation in liver with glycine and taurines, plus sodium, bile salts, sodium glycolate salts, enterohepatic circulation. Purpose: minimize energy consumption during bile synthesis. Bile in liver and gallbladder 6 to 8 times per day, terminal ileum, back to bile, liver and gallbladder through portal vein, enterohepatic circulation with terminal ileum. Factors affecting bile synthesis. Cholinergic, increased bile synthesis by bile salt, secretin, vagal simulation. Polagogs, contraction of bile gallbladder by cystocholecinin. Intestinal secretions, also known as succus entericus. Site scripts of Libercum. Types of cell.